Glucose 6-phosphate dehydrogenase deficiency is an inherited disorder caused by a genetic defect in the red blood cell enzyme G6PD that occurs most often in men. This condition occurs when a person is missing or doesn't have enough glucose 6-phosphate dehydrogenase, an enzyme which helps red blood cells work properly. Clinical presentations include neonatal bilirubinemia, acute and chronic hemolytic anemia, glucose 6-phosphate dehydrogenase is an enzyme responsible for reducing nicotinamide adenine dinucleotide phosphate or NADP to its reduced form, NADPH, in the pentose phosphate pathway. NADPH protects cells from oxidative damage because erythrocytes do not generate NADPH, thus making them more susceptible than other cells to destruction from oxidative stress. G6PD deficiency is the most common enzymatic disorder of red blood cells, affecting 400 million people worldwide. G6PD deficiency affects all races. The highest prevalence is in persons of African, Asian, or Mediterranean descent. The severity of G6PD deficiency varies significantly among racial groups. Variants producing severe deficiency primarily occur in the Mediterranean population. African populations have milder hemolysis due to higher enzyme levels. The decrease in the G6PD activity will eventually lead to the decrease of NADPH and glutathione produced using NADPH that would lead to an increase in the reactive oxygen species. Reactive oxygen species will oxidize hemoglobin precipitates, producing Heinz bodies that can cause cell membrane damage, lowering its integrity and flexibility. Cell membrane damage will eventually lead to the clinical presentations discussed earlier, like neonatal bilirubinemia, acute and chronic hemolytic anemia. Gene mutations affecting encoding of G6PD are found on the distal long arm of the X chromosome. This condition is inherited in an X-linked recessive manner and is caused by mutations in the G6PD gene. In males, one changed copy of the gene in each cell is enough to cause the condition because they don't have another X chromosome with a normal copy of the gene. In females, a mutation would have to occur in both copies of the gene to cause the disorder. Because it is unlikely that females would have two mutated copies of this gene, males are affected by X-linked recessive disorder much more frequently than females. People with G6PD deficiency do not have signs of the disease unless their red blood cells are exposed to certain chemicals in food or medicine, certain bacterial or viral infections, or to stress. The most common medical problem associated with G6PD deficiency is hemolytic anemia, which occurs when red blood cells are destroyed faster than the body can replace them.
Our red blood cells make up 40% of our blood's volume. They're made continuously in the bone marrow, looking like flattened donuts without the holes. Red blood cells contain hemoglobin, a bright red protein which gives blood its color. When we breathe, as our blood passes through our lungs, oxygen molecules attach to the hemoglobin. Then, the red blood cells and hemoglobin transport the oxygen throughout the body and release it to all our cells and tissues. When the hemoglobin molecules are emptied, they attract carbon dioxide and other waste gases, which they take back to the lungs to be exhaled. Anemia occurs when there are too few red blood cells or not enough hemoglobin. Exhaustion is the common sign of anemia.
Most individuals with glucose 6-phosphate dehydrogenase deficiency do not need treatment. However, they should be taught to avoid drugs and chemicals that can cause oxidant stress. Food to avoid include red wine, soy products like tofu, soy milk, soy sauce, and soy oil. Medications to avoid are Alaxan gel, Bengay, Omega painkiller, efficacent oil, Listerine mouthwash and pocket packs. Mothballs should also be avoided. Since G6PD deficiency is a genetic disorder, there is no cure, but the main treatment is to avoid oxidative stress. In acute phase of hemolysis, blood transfusion might be necessary. Newborn screening is a simple procedure to find out if your baby has a congenital metabolic disorder that may lead to mental retardation or even death if left untreated. The Newborn Screening Act of 2004 is promulgating a comprehensive policy and a national system for ensuring newborn screening. Aside from G6PD, several tests are done for newborn screening: congenital hypothyroidism, congenital adrenal hyperplasia, galactosemia, phenylketonuria, and maple syrup urine disease. Healthcare providers should be the primary source of information for parents regarding G6PD deficiency. Doctors and nurses should be aware of new drugs, food, or stress that can trigger the manifestation of being G6PD deficient. Midwives also has a crucial role in making sure that all babies that they deliver are tested for newborn screening. And lastly, dietitians can help parents to give proper nutrition and diet plans to G6PD deficient individuals. Being G6PD deficient is manageable, and it starts with newborn screening. Awareness and proper information is the primary role of healthcare providers in helping a child and their family in coping with this disease.